Hi, Mark. So, one aspect of your research is looking at natural regulators of epigenetics, which plays a role in cancer, right? Could you
Yes.
tell me about that? What kind of natural regulators?
Right. So this is a somehow perhaps mysterious topic for non-specialists. Indeed, basically everybody is familiar with the genetic code, the nucleotides that make up our DNA, and we all know that genetic mistakes, mutations in the DNA sequence leads potentially to cancer, for example, and also other diseases.
Hmm.
Now, over the last 20 years, basically, it became obvious that DNA contains additional master regulators that, in fact, allow activation or inactivation of the gene function. And this switch, these molecular switches, which are basically methyl groups on the C, on the cytosins, can in fact be activated or inactivated. And what works actually perfectly in a healthy human being can in fact become completely deregulated in the cancer tissue.
Okay, right. So this is epigenetics, correct?
And this is then a level of regulation beyond genetics called epigenetics.
Right, right. Okay. So a methyl group is one example of a regulator.
That is right. So again, in the healthy cell, in the healthy person, these switches are set in a very specific way.
Right.
In cancer, we have to imagine that these switches get turned on and off in a pathological way. And the idea is now to find compounds that would reset the switches in a natural, healthy way. Almost a magic trick to have a compound that is able to restore these regulatory switches. So that is one aspect of our research where we are looking for compounds. We recently published a compound called isophystylurin 3. It is a compound extracted from a sponge, which is able to inhibit the machinery, the DNA methyltransferase, so the enzyme in the cancer cell that would add additional methyl groups and as such deregulate gene expression. So by using this compound, we would somehow block this enzyme and reduce the overall load of methyl switches that are in fact attached all over the genome of the cancer cell. As such, we allow the cancer cell to become a little bit more normal and also, by the way, to die.
Okay, because that is what normal cells do and cancer cells fail to, right?
Indeed.
Yes, I
Yeah.
see. Okay.
So, on the other hand, we investigate quite a number of molecules. Some are natural, some are imitating natural molecules in part. These are the so-called inhibitors of the histone deacetylases. So this is a field where we are screening compounds and then investigating the molecular mechanisms activated by these compounds. Most of the time, these molecules lead to a cytostatic effect, so cells stop to proliferate
To stop
and then
dividing. Mm.
stop dividing, and then subsequently these cells might undergo one or the other type of cell death. So here again, we can have a very classical apoptosis, but we can also have stress-related reactions that might even lead to the so-called immunogenic cell death, where the immune system does the killing. So we have first examples of compounds that also activate the immune system, which is very interesting.